just like wasn't paying attention.
Oh, wow.
Hi, everybody. So let's get started. Okay, so this is the third lecture by So last time, so we're gonna, typically we will start with a quick review of last time. I know that there's a lot of wording, right? I'm gonna go through it quickly. It's just an opportunity for you guys to ask me any questions that you feel were addressed. If you have questions about the acid-base chemistry, we're about to go over it all again. So none of that, so hold on to your questions about that. But so a summary is sort of the most general point from the last lecture, just things to review. In biochemistry, pressure is usually taken as a constant. In a closed system, entropy tends to increase its maximum value, which is the second law of thermodynamics. However, we're not actually in closed systems. The Earth is considered kind of a closed system, right? Because we don't have that much exchange. We lose our atmosphere, right? If we were really exchanging with the rest of the universe. But generally, we're not really dealing with closed systems when we deal with cells. And so that's part of why we really need to sort of take into account both entropy and enthalpy in the Gibbs free energy. And so delta G, which is the Gibbs free energy, is the amount of energy available to do work. And so this is actually the change in the Gibbs free energy. And if it's less than 0, the reaction is favorable. If it's 0, it's at equilibrium. If it's greater than 0, it's unfavorable. And this is delta G naught, delta G naught. Delta G naught is standard state, right? But we're not at the standard state when we're actually in cells. And that's another thing to remember. Okay, so standard state, delta G naught, you're at equilibrium, right? That's where you have negative RTL and HQ. Right, and you're at the equilibrium. So I want to just point out thermodynamics is not kinetics. Okay, how fast a reaction is going to go is not related to how favorable the reaction is. You can have an incredibly favorable reaction and have it go very, very slowly. Perfect example, diamonds, right? They're still in rings everywhere, and they would be bits of graphite if it was actually a matter of thermodynamics. Okay, so the two are not the same. Kinetics is how you get to a place. Thermodynamics are the beginning and the final states, right, that really matter. It's a state function. Just note, cells are not at equilibrium, so delta G naught, it does not actually tell you what the cell system looks like. It gives you an idea of what the reaction under standard state would look like, but it doesn't really tell you whether it's going to be favorable within the cell. Right? What we're really dealing with is delta G. So the delta G naught of biochemical reactions are not always favorable. So how do they end up being favorable in the cell? Right? The temperature is not really that much of a difference, right? The pressure is constant. So what's really making them favorable? Well, usually it's a couple of different things. One is either you're playing around with how much product and reactant you have, right? So basically you're using Le Chatelier's principle and taking advantage of the fact that this is not at equilibrium to push your reaction. Okay, and the other is that you can actually couple reactions together so that you have a unfavorable reaction coupled with a very favorable one. Most typically phosphate hydrolysis, right? So the hydrolysis of ATP is one of the most typical ways in which we drive reactions forward. And so again, that's this very last part. So questions on any of this? Okay. So, since last time we seem to have a little bit of a conference with acid-base reactions, I thought we'd spend a few minutes going over something that hopefully this is the third time you've seen, right? You should have seen this in Gen Chem. You should have seen it, hopefully it's actually more than 3 times, because you should have seen it in high school, you should have seen it in Gen Chem, and you should have seen it in Orgo, and now you're going to see it yet again, right? So nothing is not repetitive. So, if we're thinking about an acid, and HA is just any generic acid, note, although we denote things A- just to make it easy to sort of balance our charges, A- does not have to be negatively charged, right? Ammonium is NH4+, and it goes to H++ NH3, right? So it does not have to be negatively charged. We just do that for convenience. So this is our dissociation reaction, right? This is the reaction of an acid going to H++ A-, so the conjugate base and a hydronium. The equilibrium constant for this reaction is known as Ka, okay? So Ka is literally the equilibrium constant of HA goes to H++ A-. Products over reactants, it's the concentration of H+ times A- over HA, okay? That's it. The peak point is quite simply the negative log of this. Okay, so when we talk about a pKa, we're talking about the negative log of the equilibrium constant for the dissociation of an acid into a hydronium plus its conjugate base. Okay, and that's it. Just to give you, this is just to go back over, in Bronsted-Lowry, we talk about acids and bases in aqueous systems, so we're talking about, and that's kind of the H+, right? That's a Bronsted-Lowry definition, where acids are proton donors, right? We're not talking about Lewis acids like metals, right, or boron, where you could actually accept electron density in, but you don't have a proton. We're really talking about Bronsted-Lowry acids, where we're defining it by HA goes to H plus plus A minus. So that's a Bronsted-Lowry definition of an acid, okay? Okay. So this is just something that some of you may, if you've ever taken organic chemistry with me, you may remember. And this is just a basic pKa chart. And it's to give you a little bit of a grounding in approximately where pKa's lie, right? So remember, it's the negative log of the Ka. So if your pKa is negative, right, it means that your Ka is very, very large, okay, because log is base 10. So a negative 9 pKa means that your Ka is 10 to the 9th. Okay, just to calibrate you on this. It sometimes gets confusing, right? pKa goes very low, but it actually means your equilibrium constant is very high. It means that you're dissociating into the acid quite readily. Okay, you're dissociating the acid into the hydronium and the conjugate base quite readily. So for example, HBr is a very strong, it's the strongest acid on this list. It's a very, very strong acid, right? Hydrobromic acid. Br minus is the conjugate base. Its pKa in water is -9. Okay, it means if you put this in water, you're going to have 10 to the 9th H plus times A minus over HA. Okay, so you'd have to take the square root of this to be able to actually, it's like 10 to the 4.5 A minus, right? Okay, so sulfuric acid, again, very, very strong, -3. Hydronium, right? So if you have H3O plus, protonated water in water is -1.7. Okay, acetic acid is about 5. This is actually kind of a good number to know, so 4.75. You don't have to know the exact number, plus or minus a little bit, it's 5. Okay, that's what you really need to know. The reason that that's a good number to know is because that carboxylic acid motif is one we see quite often when it comes to proteins, right? So when we start talking about protein structures, carboxylates, carboxylic acids, the amino acid, the acid part of an amino acid is a carboxylic acid, right? So that pK is kind of a nice benchmark to kind of tell us where things are in this hierarchy. Conversely, when we talk about amino acids, the amino part is usually an amine, and in water, not really in water, in buffer, I should say, because it's really buffer that we're talking about, right? In buffer, it's typically, that's buffered at physiological pH, it's typically protonated, right? And the pKa of ammonium is about 9.2, okay? And this is an example where the A minus isn't actually negative, right? Conversely, the pKa of water is about 15.7, okay? If you divide that by 2, that actually gives you your pH, right? So the pKa of T-butanol is about 17. So again, because you have electron donation, because when alkyl groups have a little bit more ability to donate, right, to the oxygen, you actually have it. It's a little bit more basic, right? A little bit more able to take a proton than to give one. Okay, so that's about 15. Oh, sorry, this is about 17. Ammonium. So this is ammonia. You notice ammonia going to NH2 minus, right, is 38. So don't confuse these two. This is a very common mistake. People confuse the pKa of ammonium and ammonia. Okay, ammonia is going to NH2 minus. Think sodium amide, incredibly strong base, right, going to go to ammonia. Okay, that's completely different than thinking about ammonia as a base, where then you're dealing with the pKa of ammonium, right? So, and then, of course, if you were going to try to deprotonate, this is a very, you can make lithium salts of these, and this gives you a little bit of an idea of how incredibly basic they are, but, you know, you typically don't deprotonate methane, right, because it's an incredibly bad acid. and the pK is around 50. This is sort of the gas phase assumed pKa by extrapolation. So, okay. So, coming back to this, so again, Ka being this, and pK is the negative log of the Ka. Let's get into how we get to that Henderson-Hasselbalch equation, right? So we got to that equation, and I kind of magically put it up there, but I think it's sometimes good to sort of step through to sort of see how we actually get there, right? So we know that the pKa is negative log of the Ka, which I've now written out as H plus times A minus over HA, right? Products over reactants. So if we just do a little bit of simple rearrangement, logarithmic math, right, we know that if you think about it, H plus, you can actually rearrange this to be H plus times A minus over HA, right? And if you remember, when you have a multiplier inside the parentheses of a log, they're actually just adding the logs, right? So that means that this is negative log H plus the log of A minus over HA, okay? Which, if you take the negative charge through, or not negative charge, the negative through, right? So it tells you I'm a chemist. I called it a negative charge. Okay, so if you take the negative value through, this is now negative log H plus minus negative log A minus over HA. If you take this part right here and put it on the other side, it's pKa plus the log of A minus over HA equals negative log of H plus. And you might recognize negative log of H plus as pH, right? And this is essentially the Henderson-Hasselbalch equation. It's just math, okay? It's not some sort of magical thing that you have to derive by any sort of magical means. It's basically math, okay? It's solving for Ka and solving for the negative log of H plus by using the fact that the equilibrium constant is H plus times A minus over HA. That's it, okay? What does that give you? So that gives you this, right? What does this do? This relates acid-base equilibria with the pH, okay? It tells you how much dissociation, how much A minus over the conjugate acid you get in a buffer of a specific pH, okay? Or a solution that is of a specific pH. Okay, so when the pH is equal to the pKa, right? So when these two things are equal, the log of this needs to be 0, right? And the only way for that to be true is to have A minus be over HA, right? Because the log of 1 is 0. So that's essentially how we get to this part of the equation, okay? So when pH is equal to the pKa, the concentration of A minus is the same as the concentration of the, it's not, I wouldn't say it's an equilibrium point because it's not the right word. The reaction's not at equilibria, right? It's the point of equality between the dissociated, conjugate base and the acid, okay? The reason it's not an equilibrium point is because that's not the KEQ, right? That's not the equilibrium, that's what Ka is, right? And so I guess it is kind of the K. I take it back because of the fact that the pH is equal to the pK, it is actually the equilibrium point between these. So I take that back. Okay. So in proteins, there's a reason. So actually, just talking about this generally, there's a reason why we want to look at it as this rearrangement. So if we have the pH of a solution and we subtract the pKa and raise it to the 10th power. So we're basically just taking 10 to the pH minus pKa. That will actually give us this dissociation at any particular pH. Why does that matter? That matters because we might want to know in a buffered solution how much how much dissociation we're going to have. And this is going to matter for proteins because when we think about what's happening inside the cell, we're not in water. And let me say that this original slide was wrong. Can anyone tell me what was wrong about this original slide? And I didn't catch it. Rahini actually caught it. It was actually a mistake that was carried through from a slide that my colleague had given me, and I didn't look at carefully enough. Nope, it's still 99% ionized. What's the problem? The original slide said A minus over HA in water, right? What's wrong with that? Because if you were to take acetic acid and dissolve it in water, what would happen to the pH? It would change, right? So in that case, we wouldn't be talking about a pH of 7 anymore, right? So that was what was wrong with the original slide. If I had just dissolved acetic acid in water, the pH would be far lower, right? And so we would no longer be talking about this. So that slide was incorrect, okay? And you'll notice in the new one, I have actually pointed that out by changing it to in buffered solution, pH 7, right? Because this is accurate to what you're looking at. And the reason that it's important it's a buffered solution, the reason we talk about things in buffered solution is because your cells are buffered solutions, right? When we think about your cells are not water. They are not bags of water, okay? They are bags of many, many things. They're not even bags. They are full of many things. It's actually kind of a hydrogel environment, but part of it is that there's a lot of buffering agents in there, mostly phosphates and carbonates, etc. Right? So your cell is buffered to a specific pH, and it does its best to maintain that pH by a lot of different means, some of which are just simply straightforward inorganic buffering, right? And some of which are actually ion pumps that maintain you at a specific pH, right? But the combination of all of that is that your cells are buffered. So when we talk about, pH minus pK and talking about ionization, we're really talking about the pH of the final solution. Right? So if you're dissolving in water, your final solution is not pH 7. Okay, it's some other pH that you'd have to figure out. Okay, so in a buffered solution pH 7, the concentration of A minus over HA is 10 to the 2, which means there are 100 molecules of acetate to 1 acetic acid. So this is now actually an accurate slide. So it's 99% ionized in approximate, okay, it's a little bit more than 99% if we do a pH 7.2 buffer, but it's approximately 99% ionized in the cell. Okay? That makes sense to everybody? Okay. So getting back to physiological pH, right? So the physiological pH range, the real one is between, the book says it's about 6.5 to 8. The truth is in the vacuoles, if you look at this Leopis PNAS paper, they estimate that it's approximately 5 in the lysosome by the current methods that they've been able to use. So in this paper, they actually do pHing of the various organelles using various reagents that people have developed to do exactly this, right? So people have used genetically encoded sensors to look at pH in organelles. They've actually made small molecule sensors to look at pH in organelles, and these are the approximate numbers. And they're approximately what people think it is. So cytosol is everything that's not these membrane-bounded structures, right? Cytosol is about 7.2, and that's what we normally consider physiological pH. So when people say physiological pH, they typically are referring to a pH of 7.2. They're really referring to the pH of the cytosol. Why? Because when you lyse cells, the vast majority of stuff that you get are cytosol, right? So that's probably why that's always been considered physiological, right? The lysosome and vacuoles, which are places that degrade intracellular materials, right? They're part of your degradation pathways. They're more acidic, and there are various enzymes that actually take advantage of this to break apart your proteins and various other molecules in your body. Okay. The Golgi, which is where carbohydrates are made, and we'll get to that a little bit later in the semester when we talk about carbohydrates, is also slightly acidic. And then your mitochondria, which are your little, I don't know where they are. Here's the mitochondria right here. They look kind of like bacteria because once upon a time they were. They're the little powerhouses of your cells. That's where ATP actually gets made. It's actually a bit basic, okay? And people actually take advantage of these, as I said in the last lecture, to do various things. So people often take advantage of the fact that when you have endocytosis of, say, a drug from the cell surface, if it's coming in through what's called a passive mechanism, where it's getting endocytosed in just through sort of sampling of the environment, it often goes to the lysosome, which is then more acidic. And people have done all sorts of really cool designs to actually make it so that drugs will break off from a carrier at that point to be able to enter the cell, and used it as cell entry mechanism. So people have actually used these pHs to do some kind of cool chemical tricks. Okay, I want to point this out. This is mostly just for your knowledge, which is that we often take the pH of water to be 7, right? But when we talk about the pH of water to be 7, we're really talking about, so it's half the pKw, right? And when we talk about that as 7, we're really taking it to be 7 because of standard state, right? At the standard state, which is temperature about 25 degrees, right? Temperature of 25 degrees, the pKw is approximately 14, and so that's why we take it to be 7. And I just wanted to point out that at the 37 degrees where your body hopefully is, right? That's like the normal human body temperature, right? So your 98.5 is about 37 degrees Celsius. So at 37 degrees, the pKa of water is actually slightly lower, right? So it's more in the 6s. And so that's just something that you should be aware of. This has to do with Van Hoff's equation. You don't need to, I don't want you to ponder it too much, but I just want you to be aware that, equilibrium constants are temperature dependent, and that includes the equilibrium constant for water dissociation, right? And so it is slightly more acidic. So water is by our, metrics, slightly more acidic when it's warmer, okay? Okay. So let's talk a little bit about what acids and bases kind of look like in proteins. We're going to spend an egregious amount of time on this in not too many more lectures after we go through nucleic acids. So I'm not going to spend a ton of time now, but I just kind of want to give you a little bit of an idea what's there, and what that looks like in terms of pK and pH. So the C-terminus, so I told you these are amino acids. So the acid part of a peptide would be the carboxy terminus. Has a pK of about 3. You can see that, and obviously it's a little bit lower than your typical carboxylic acid on acetate, which is about 5. On the other hand, when you have these aspartate or glutamate side chains, you'll notice here the pK is a little bit higher, right? A little bit closer to our typical carboxylate of acetate, which is kind of our model compound. Okay. Histidine, which is an amino acid we'll go into in probably gruesome detail, which is an imidazole, has a pH of about a pKa of 6.6. And this is actually really important. This is a very special amino acid, and it has some really interesting properties catalytically because it hovers right at that point of physiological pH, right? Which means that some of it's protonated and some of it's deprotonated. And that actually has some important consequences in terms of its ability to act in enzymes in catalysis, right? Because it can act as an acid and as a base. And so because of that, you often see it in enzyme active sites. And we'll talk about that more when we get into proteins.
Why are the two first groups, the aspartate and glutamate side chains, and just the typical carboxyl group, slightly different pHs, pKa?
So the pKa has to do with many things. I mean, so we're just showing the functional end of the molecule, but the carboxy terminus, as when we get to amino acids, we'll draw it, but it has the C alpha carbon, and so it will partly depend on what is at the C alpha for exactly what that pKa is. But the difference is like the aspartate and the glutamate have alkyl side chains. And so those alkyl side chains are more electron donating, and so that may be part of why the pKa is higher, right? So it's a little bit less likely to want to ionize, right? So the pKa will be higher if it's less likely to want to ionize. Having donating groups would make it so. So, and this carboxylate probably, that's why it's an approximate. I suspect it changes based on what your terminal amino acid actually is, right? If it's a serine where you have a hydroxyl group next door, it's an electron withdrawing group, it's probably lower. If it's an alanine where you have a CH3 group next door, it's probably slightly higher, right? So, and then like the N-amino terminal group is sort of the same way. So the terminus, the amine, right? So typically they show here as the ammonium because it's typically protonated at pH 7 or more of it is. You have it's about a pK of 8. Cysteines are typically still protonated, but they can have a thiolate, and this is a pK of 8.7. Again, this has some very important chemistry that we'll talk about as we get to proteins. Tyrosines are about, this actually follows quite nicely with phenol. Phenol has a pK of about 10. Tyrosines have a pK of about 9.8. Very similar structure to phenols. The lysine side chain, again, this is an amine. Again, this because lysines have a lot of alkyl groups here, they're more electron donating, the pKa is actually a little bit higher. Okay. And arginine is actually quite a bit higher, it's at 13. Does anybody know why? What's different about arginine?
Is the bond has all of those like NH3 plus?
Yes, it's the resonance, right? If you actually look at arginine and you notice it's got these dot, and that's because that's actually a hybrid resonance structure. And we'll go over what that actually looks like in terms of arginine, but but this particular structure is very, very resonance stabilized, and that's part of what makes it such a strong base, and it's one of the strongest biological bases that one sees in terms of the proteins, in terms of amino acids. Okay, I think it's the strongest base as an amino acid. Okay, so going back to the Henderson-Hasselbalch equation, just pointing out, when the CH is equal to the pKa, again, this is where you have the equivalent amount of conjugate base to conjugate acid. Why do we point this out? Partly because when we think about proteins, part of what we think about is how ionized any particular amino acid is in that protein, right? And that can be very, very important, and it contributes to what we call the isoelectric point. The isoelectric point is actually used in proteomics to help separate out proteins that can have different compositions. Okay, that may have the same molecular weight but have different compositions of ionizable groups. Okay, so what is an isoelectric point? So to have an isoelectric point, you have to have an amphiolite. So an amphiolite is something with both acidic and basic groups. All proteins are amphilites because they have amino acids, right? That by itself, the amine and the acid gives you an acid and a base, right? Or base and an acid. So you instantly have an amphiolite in any amino acid, okay? Which means, of course, all peptides are amphilites just by their very nature. The isoelectric point is the pH at which the average charge for every form of the molecule is 0. So it is the pH at which the molecule is now neutral. That does not mean that every amine is deprotonated and every carboxylic acid is protonated, right? Because individually they're not. It's talking about the total balance of charges within the entire molecule, okay? So that's what the isoelectric point is. It's when all charges are equal to 0. That's actually really useful because one of the things that people do in electrophoresis, and we'll talk about this more when we get into proteins, is they'll run things by their isoelectric point. Okay, so they'll run things on a pH gradient, and those will stop at their isoelectric point, and then they'll run them down, and that'll actually allow them to do separations of really complicated groups of molecules. And that's actually what part of what's called 2D gel electrophoresis. Okay, so at pH less than the pi, a molecule is positively charged, okay? And at a pH greater than the pi, the molecule is negatively charged. So here's an example of aspartic acid. It's got two acids and a base on it, right? If we look at low pH, if we look at low pH, where the pH is less than the pi, you'll notice that the acid is protonated, and so the base is protonated, and so are both of the acids, right? So it's positively charged. When you're higher than the pi, now the base is deprotonated, and so are the acids. So you now have negative charge. Exactly at the isoelectric point, which is right about here, it's kind of a little wobbly because it's basically the equilibrium point for this, right? Where you have kind of an average, like a hydrogen that's kind of going back and forth between these two, would be the isoelectric point for this. So this is the titration curve for aspartic acid. So looking at something that's more like a peptide, you can see like in a peptide, each one of these at physiological pH of 7 is going to be a little bit different in its deprotonation state, or protonation state, right? So tyrosine is about 13%, 0.13% deprotonated, but glutamate's about 98% deprotonated, right? And so all of these charges together have to equal 0 for you to be at the isoelectric point of this peptide. Okay. And so just to show that in a different one, so here where you have a total charge of 0 is where you are now, is that pH is your isoelectric point. So that's the pi of it. Okay. So getting back to acid-base chemistry. Questions on isoelectric point? It's basically the point at which all of your charge is 0. Okay, it's the pH at which all the charge is 0 on average. Okay. In a molecule where you have an uneven number of charges, it's the average of all of the molecules, right? So it's the ensemble of all of the molecules. The charge is 0. right? Not each individual particle. Okay, so getting back to acid-base chemistry, we're going to talk a little bit about buffers, right? Because that's what happens inside the cell. We're buffered. We have pH 7.2 of the cytosol. It's not water, right? If you dissolve a carboxylic acid in it, it doesn't automatically make it, pH 3 or whatever the pH would be if you just dissolved any, depending on how much carboxylic acid you added, right? You're buffered to be within a very specific range. And so let's talk a little bit about this buffering capacity, right? And what exactly is a buffer? How does this work? So again, in the physiological range, we're talking about this range right here, 6 to 8 is about where we normally are for things that are inside of a cell. You can tell just by looking at what is in this range, what one of the major buffering agents inside the cell is going to be, right? Which is dihydrogen phosphate, right? So this is actually one of the major buffering agents inside of a cell. We have a lot of phosphate buffer basically inside of our cells. And you also get some buffering from carbonates as well, but that's kind of a different issue. But this is a major buffering agent, and you can see it has a pK that's just about in the middle. So, what is a buffer? If, again, going back to the Henderson-Hasselbalch, so if A minus is equal to HA, this is where pH and pKa are the same, right? So, what does that mean? It means that if you say that you have acetic acid and you dissolve it in a solution of water. So you start out and you're just in water, right? And so the pH is quite acidic, right? This is for a set amount of acetic acid, let's say 1 equivalent of acetic acid in water, okay? So you're keeping the acetic acid amount constant. If you then titrate in hydroxide, you'll come to a point where you will actually, where the slope of the line is not sharp, okay? And the reason is because you're in this buffering region. You're in a region where you have actually 2 competing reactions. So your equilibrium is not just set by this reaction, it's also set by the hydroxide to water reaction, right? So you have 2 competing reactions. And the buffering region for any acid is actually 1 pH unit plus or minus from its pKa. So for something like HCl, there basically is no buffering region, right? Not on this scale, okay? Because the pK of it's negative something or other, right? So you add a little bit of acid. I mean, like as you add equivalents of acid, it's going to go completely up or completely down. It has no realistic buffer. But something that is in the physiological range, being buffered with something whose pH, in this case, or pKa, is close to it, like in water, right, is actually has a buffering range. It's, as I said, it's like plus or minus 1 pH unit from the pKa. So how does that actually work? So this is if you think about the competing equilibrium reactions, right, this is actually what that looks like. So your acetic acid is getting either protonated or it can get deprotonated. So this is going to water. This is the H+, this is going to water. So as you titrate in more OH-, you're going to the acetate. And you have these competing reactions, right? You have the Ka, which is your dissociation of your acid, right? And you have your Kw, which is your dissociation of water into H+ plus OH-. Okay, and these are the 2 competing reactions that you have. And because of these 2 competing reactions, as I said, you have this buffering region when you use hydroxide. Okay, because hydroxide is part of because you're in something whose natural dissociation constant is in the same range, right? Is in that pH 7 range. Okay. So nature uses hydrogen phosphates and also carbon dioxide carbonates to buffer. Typically inside the cell we see hydrogen phosphates, but one of the things that we see is but nature tends to use carbonates, for example, to acidify things. Okay, and we see that with CO2 exchange. Okay, and that's part of why nature uses carbonates that way. Okay, so what is a buffered solution? So obviously, when we talk about buffered solutions, you could have some different solvent that has a dissociation constant that allows you to have a buffered solution of something else. You could have a different competing reaction. But when we talk about it, we're really talking about water, right? Because we're in biology. We're not in some weird silicon solution of, you know, something else on some liquid planet that has like weird pressure, etc. Right? We're talking about water. Our life is in water. And so we need to have things, you know, you can't buffer something that has a pKa of 20. You can't buffer something that has a pKa of -1, right? That has no buffering capacity. You can only buffer things that are, as I said, around the physiological range, between 6 to 8. Okay, so that's kind of your buffering range for water. Okay, because you have to have a competing reaction with a Kw. Right? And you can't have that if your pKa is really wildly one way or the other. So buffered solutions have this property that they are able to minimize the change in pH following the addition of an acid or base because the conjugate acid and conjugate base of the buffering compound are present in sufficient concentration to combine with the H plus or OH minus and neutralize them. Okay? And that's why you can't do it for something that has a pKa of -1 because you have way too much H plus to be able to neutralize the OH, you know, to be able to have it neutralized by the OH minus you're adding in. Okay, like it's too dissociated. Okay, you have no capacity. So they function because the pH of a weak acid or base solution is least sensitive to added acid or base near the pKa, i.e. where the conjugate acid and conjugate base are both present in nearly equimolar concentrations. Okay? Make sense? Okay. So just to give you an idea, these are common biological buffers used in molecular biology. This Hepis and Tris are actually probably the most. This Tris as well are some of the most used buffers. And their pKa's, as you notice, are all in that 7-ish range, right? So this has a buffering capacity, you know, about 7.2 to 5.2. You would use cafenylic acid if you're trying to buffer something to be a little bit more acidic, right? You would use this Tris if you're trying to buffer something to be a little more acidic. If you're trying for something that you want to keep around 7.2, you're probably going to use Hepis or Tris, right? Tris is actually if you're going to go a little bit more basic, okay? So you can choose your buffer to sort of tune your system, right, to give you a little bit more capacity towards the pH that you actually want to try to maintain, okay? Okay, so that's pretty much it on acids, bases, Henderson-Hasselbalch equation, and buffers for now. Questions on any of that? Okay, so now we're going to actually talk about some molecules, some different molecules, different than acid and base and pKa. We're going to start out talking about the central dogma of molecular biology. So what is a central dogma? So first of all, let me just say that dogmas are completely meant to be overwritten, and the central dogma has already been overwritten. But it provides a nice framework for sort of learning biochemistry and molecular biology because it's sort of the traditional way in which it tends to be taught. So this actually comes from work from Watson and Crick, where there is this quote actually from Crick that says, the central dogma of molecular biology deals with the detailed residue-by-residue transfer of sequential information. It states that such information cannot be transferred from protein to protein or to either protein or nucleic acid. So the idea was that information basically only flows one way in biology, that it goes from DNA, which could be replicated and replicate genetic information to make more DNA. It could be transcribed into RNA, right? The RNA can be translated then into protein, and this can then catalyze various reactions to make other biomolecules. But the central dogma kind of argued that, you know, RNA couldn't contain genetic information that would then lead back to DNA, right? Proteins couldn't control DNA genetic information. All of this has been overturned, and we'll talk much later in the semester after we've gotten a little bit more under our belts in terms of basic biochemistry about some of the ways in which this is no longer true. For example, we now know that proteins can catalyze, you know, histone methylation, which can turn off genes and make it so they can never get turned on, right? Which means that actually then does feed back into regulation of what happens at a genetic level, right? You actually can inherit histone marks, right, from proteins. And so this is a completely different way of thinking about it. And people still don't, this is part of what's called epigenetics, and people are still trying to figure all of this out, right? Turns out, and this is one of the great things about science, which I personally completely love. So when I was a postdoc, I postdoc for James Rothman, who won a Nobel Prize for his work in snare proteins in the Golgi. And in my lab was a fellow postdoc named Frank Carlotti, and the human genome map had just come out. Okay, so this was 2000. I'm dating myself, but this is 2000. And the human genome map had just come out, right? And Frank said to me, he's like, that's it. There's no point in being a scientist. We're going to know everything in like 5 years. Why bother? And I said to him, I'm like, nope, we're going to find out that we know nothing. And all I have to say is I was totally right. We know nothing. We know nothing, and it's awesome, right? Because what more fun is there for a scientist than to figure out all the stuff we don't understand, right? There are so many questions we still have. And so I will teach you what we know currently about biochemistry, but it's a living science, right? Our understanding of even basic things like how chemistry really informs our understanding of structure is still evolving, you know? So I will teach you our current knowledge, but you should just know that, you know, chemistry is a living science, biochemistry is a living science, you know, science is still very much a living thing. If we understood everything, we wouldn't have so many fewer problems, or maybe we'd be much more screwed up. I don't know which it would be, right? But either way, I know that we're not there yet. So this is the central dogma, and like many dogmas, it has already been overturned. However, it does provide a nice sort of way for us to think about our macromolecules. So we're going to start out talking about DNA, and we're also going to talk about RNA, so we're going to talk about these two together. And let me say that here is one point where you really will need to pay attention to the slides, because although this book has many really good points, it's not very thorough when it comes to a discussion about DNA and RNA. I will say that that chapter is a little bit skimpy. So there'll be more information in the slides than there will necessarily be in the book. Okay. But I have actually, again, written it out in the slides, so it should be pretty clear. So, and then from this, we'll start to talk about proteins, and then eventually enzymes, and metabolic pathways, and then genetic regulation. So we'll talk about where this is now. Okay? Okay, so first I just want to sort of show you a picture of DNA and RNA structure. And you probably can already notice something really different from the two of these, right? DNA, I'm showing you a helix. And we're going to talk all about the nature of the helix. You probably have seen this DNA helix motif in many different places, right? It's become so ubiquitous that pretty much people know, oh yeah, DNA, it's a helix. Right? So DNA is a two-stranded helix. It actually has two different strands of DNA that are coming together in a complex, right? And this is the typical structure. This is actually the Watson-Crick structure of DNA. RNA, this is actually all one RNA strand. It's single-stranded, not double-stranded. And you notice that it's folding into a much more complicated structure. In fact, this is a transcript RNA. So this is a tRNA that's involved in protein translation. Okay, this is only one of the myriad of structures that RNA can actually fold into. So instantly you can see one difference between DNA and RNA. DNA has a very sort of narrow molecular space, sort of structural space in which it tends to fold. And although there are some really cool DNA nanotechnology molecules that people have managed to make, in general, all of it basically takes advantage of this helical formation. Okay. RNA, on the other hand, is much more flexible, has a much larger folding space, okay? And we're going to see examples of why this is. So RNA much more flexible than DNA. Okay, so just showing this another way. Here's in DNA you have two strands. So this is showing it with the bases just as these color-coded bars. Right? And this is supposed to be the phosphate backbone. We'll talk about this in a minute. And here you can see in DNA you get this nice duplex. So DNA is a duplex. RNA is single-stranded and forms into these really complex molecules where parts of it look like duplexes, right? And parts of it don't. Okay? So let's take a little bit of a look at what those strands look like at a chemical level, right? So in DNA, we have this deoxyribose ring, and we'll talk about this in more detail in a minute, that has a nucleoside base, and again, we're going to talk in more detail, so this is the really, really rough draft of what we're looking at. And you'll notice that they're all connected by a phosphodiester backbone, right? Highly negative charge, lots of phosphate, so there's a single phosphate in between the hydroxyl at the 3' of the ribose and of the 5' of an adjacent ribose. Okay, so this is one sugar, This is another sugar. Okay. Now, RNA looks exactly the same as DNA, except for one critical difference. Actually, there are two differences, and we're going to talk about this, but there's one that you can see in this picture, right? And that's this hydroxyl group here. And in fact, this hydroxyl group is part of what has all of those incredible structural implications. Okay, and we'll talk about that in a little bit. Okay, so again, this is one of the main differences between RNA and DNA. DNA is 2' deoxyribose. Okay, so this is what's called the furanose form of that carbohydrate. You might remember that as a carbohydrate, this is actually, if you opened this up, it's actually an aldehyde, right? This is a closed acetal. Okay, actually this is a hemiacetal because it doesn't have a different. If it had a second R group here, it would be an acetal. This is a hemiacetal. Okay. When the base is there, it's really a hemiaminal, but because it's connected, we call it a nucleoside. Okay. So when the base is there, this can't really open. In this form, it can open and close. Okay. But when the base is here, it doesn't actually form the open chain, right? It's stuck in the closed furanose conformation. Now, you might notice that this looks kind of like a pentane, right? And in fact, we'll talk a little bit about how that whole envelope structure, you guys remember the envelope where it flips up? That actually has a role in DNA structure, okay, and in RNA structure and the differences between them. And we'll talk about that in a minute. You thought you could forget all about pentane conformations, I bet. But no, biology uses them. Okay, I know. Okay, so a difference with ribose and deoxyribose is deoxy again has the hydrogen, and ribose has this 2' hydroxyl. So in the nomenclature of DNA, the nucleobases are numbered 1, 2, 3, 4, 5 for the atoms in their rings, and we'll go over that in a few minutes. And the ribose is always given the prime number. Okay, so 1' is the anomeric carbon of ribose. Okay, this is C2'. This is the C2' hydrogen. This is the C2' hydroxyl. Okay? This is 3', 4', 5'. When we talk about 5' to 3' in DNA, we're talking about going from this position to this position. Okay? And that's how they're linked. This is where the phosphate is. There's a phosphate that would go here, and it would connect to this hydroxyl on a different nucleoside. Okay. So this is what the bases look like. And yes, you have to know the structure of the bases. This is always a question. Do I have to know and be able to draw the structure of the bases? I hate to inform you of this, but absolutely. There are many things that we'll be able to learn from first principles and work our way up. Some things you just have to memorize. This is one of them. Okay, so know these. You're also going to have to memorize the amino acids, just so you know. Okay.
What do those arrows represent?
I'm going to tell you that in just a minute. That's a very good question. The arrows actually will really come in handy when we start talking about Watson-Crick base pairing. What they actually represent is places where you have a hydrogen bond donor and a hydrogen bond acceptor. So the red arrows are acceptors, and the blue arrows are donors. Okay, and this is a nice image from the Molecules of Life. I like this one because I think it really kind of shows the hydrogen bonding capacity of the different bases in a really obvious way. So I sort of like that. And it connects again to our hydrogen bonding motifs, right? Yes, we talked about how hydrogen bonding is involved in specificity. Hydrogen bonding. We're going to see a lot of this, okay? So there are two classes of bases, purines and pyrimidines, okay? Purines are these bicyclic compounds. Pyrimidines have just this structure, have 2 nitrogens in an aromatic ring. Okay, so you notice that the actual aromatic ring doesn't quite look like this. It depends on what there is. So in adenine, we have, again, let me just say the base is actually connected here at what's called the 9 position of purine. Don't ask me why it's numbered this way, okay, because I don't really know. I think it has to do with, I have actually no idea. I don't know why it's numbered this way, but they numbered it, so this N is 1, okay? Go figure. The place it connects to the ribose ring is actually what's called the 9 position. So it's 1, 2, 3, 4, 5, 6, 7, 8, 9. I know that it's like you don't even go around the whole edge of it. Okay, and it's because it's two. You can think of it as 2 aromatic rings sort of fused. It's 2 fused rings, right? It's a fused ring system. So let me point out. Well, actually, let me ask you. What can you already tell me about the nature of these bases? Is there any sort of special chemical property that these bases might have? Well, they have the hydrogen bonding accepting and donating, that's true, but what else is true about them? They have high stacking, but what is it just, is it a normal pi bond? Like, what is there about them? They're aromatic, right? Now, so think to yourself, why are they aromatic, right? I mean, if you look, it's not necessarily instantly obvious that guanine is aromatic, right? But it is. Why? Why is guanine aromatic, even though, you could argue that there are 2 electrons here that are conjugated in? Well, think about it, right? This is actually aromatic, but why? So, you want to answer? Yeah.
Well, you have the lone pair on the nitrogen.
Well, it is true that there is a lone pair on the nitrogens, and that is part of it. So how does it end up getting aromaticity?
The oxygen?
A little bit. It's the resonance structures. Remember, if something is aromatic in the resonance structures, it's aromatic. Now, let me say, it's not as aromatic as something that would be in its resonance structure. Let me just say, the aromatic resonance structure, although it is a major one, has one small issue, which is that it is also very charge separated, right? Because if you delocalize, for example, this lone pair into this ring to make it aromatic, these electrons go up to nitrogen. This is negatively charged, and that's positively charged, right? So it is a charge-separated structure. So that means although it is aromatic, the aromaticity is perhaps not as much of a stabilizing influence as something like benzene, right, where everything is completely shared evenly. Because there is this whole issue of electron separation of charge, right? So where you're actually then going from a neutral molecule to a charge-separated one. And that costs some energy, okay? So it is aromatic. The resonance structures tell us that it's aromatic. The biology tells us it's aromatic, but it's not as aromatic, but its relative aromatic stabilization isn't as much as you might think for like a molecule that's, like benzene or naphthalene or one of those hydrocarbon molecules that don't have this charge separation issue. Or purine, right? Okay. So again, we have this purine, and so in this purine, so this is adenine. It has this NH2 group here, versus guanine. So guanine has this trinitrogen. That's kind of how you remember guanine, because this is actually the sort of key characteristic. And this carbonyl group here. So it's an amide, and then the amino group, and then it goes into this nitrogen. So that's the difference between guanine. You can see that there's definitely a different hydrogen bonding capacity for guanine and adenine. Cytosine has a carbonyl group and an NH2. Okay, and the numbering here is 1, 2, 3, 4, 5, 6. So carbonyl, NH2. Uracil, on the other hand, has 2 carbonyl groups, and let me say that uracil is only found in RNA. Okay, so uracil is an RNA base, not a DNA one. And thymine is the same as uracil, but it has this methyl group, and thymine is only found in DNA. And we'll talk a little bit about why that is. Okay. So really quick practice, given that this is adenine and it's connected there, draw the deoxy, draw deoxyadenosine, i.e. the nucleoside base. So I want you to attach it to that ribose, that deoxyribose structure. Draw what you think that will look like. Okay? So given that this is adenine, right, and we know what ribose looks like, and it's 2' deoxy, draw how that all comes together in your molecule. I'm going to give you guys, this is your extra credit. I figured I'd give you a little practice drawing these nucleosides. just to give you a little, just to remind you, this is what deoxyribose looks like, right? So you're going to put the base there. Just as a quick reminder. I don't know which one you need more. They'd probably be adenosine. So I'm going to go back to the adenine. Okay, so here's the adenine. Give you guys one more minute. We'll move on.
Oh, you started over?
See that none of it was actually.
Yeah, that was very sad. What is it
No, sure.
Oh, she was just going to.
Okay.
So
So this is what it looks like.
Oh.
And again, whatever you've drawn is fine. You don't have to have it perfect. We're not grading you on this. This is actually so that we can get a sense of, you know, your drawing abilities and then just getting a little bit of sense of things that are easier or harder for you. This is just practice. So this is actually the D-adenylate because I show you here the nucleotide. You call it a nucleotide if it's phosphorylated. I didn't ask you to draw the adenylate. I asked you to draw the adenosine, right? Deoxyadenosine. So the nucleoside. So the nucleoside is a sugar plus base. The nucleotide is all of that plus the phosphate. That's just the difference in that. So here's the base. This is adenine. Here's that 9 position. The hydrogen is gone, right? Because otherwise you would have this positively charged. It would be kind of a mess. It would probably break off quite easily because the oxygen would open it up and you'd remove the base, right? That kind of gives you an idea of things that could happen to this. So, and then you have what's called a glycosidic bond between the nitrogen and the anomeric position, okay, of this closed Uranus ring. Okay. So, getting back to just RNA and DNA-based structures, I just want to point out something that we're going to talk about much more when we get to the actual structure, the helical structure of DNA, when we talk about why it is stabilized. And we've touched on this already, but we've talked about packing, right? We've talked about dipole-dipole packing interactions and pi stacking. But I want to point out that when we think about A, G, C, and T, and also U, but here I've just got the pictures for A, G, C, and T, and you look at the electron density maps, they're not even, right? And I just kind of wanted to show you, these have permanent dipoles in them, right? And you can imagine that as these things stack, that some interactions are going to be stronger than others. And in fact, that's exactly what we see. And when we get into what stabilizes the helix, we'll talk a little bit about how differences here can actually change the melting temperature and the stability, essentially the thermal stability of DNA. Okay, and that can have impact on, you know, how easy it is for an enzyme to say unwind it, right? To be able to polymerize things or to be able to hybridize the things. And we'll talk about all of that as we get further into the structure of DNA. I mean, right now I just wanted to kind of introduce the concept that all of these rings do not look the same at an electronegativity level, right? If we're looking at electron density. Okay, so I also wanted to point out that under certain circumstances, these can actually tautomerize. So just to remind you, tautomerization is something where you have two forms that are differentiated by where the hydrogen is. It is not a resonance structure. In a resonance structure, the atoms do not move, Okay, and also resonance structures are not equilibria. They're just a way of thinking about the overall structure of a molecule, right? All of the resonance structures contribute to the structure of a molecule. They are not individual, they are all the same molecule, okay? This is actually an equilibrium process where you can actually get, the hydrogen is changing, you actually, and this can be catalyzed by things, where now what's happening is you're getting, this would get protonated, and then, so this would get protonated, and then these electrons would come here and go up to oxygen, right? And that's how you would get to the enol. Okay, so this is, so both guanine and also thymine actually can tautomerize to the enol, and you might remember your mechanisms for that. And adenine and cytosine actually tautomerize to the imine. So in these cases, the hydrogen, this nitrogen can get protonated, and then these electrons come down and kick those electrons to that nitrogen. This is actually quite important because this is not necessarily stable to hydrolytic attack, right? And that will actually be something that we'll talk about in a minute. Okay, so another thing about these bases, they are aromatic, and like many aromatic molecules, they absorb UV light. Okay, so these are actually, can absorb UV light. They absorb UV light at about 260 nanometer. Why is that important? Because when people, if you ever work in a molecular biology lab and you're trying to test your DNA, you'll often take an A260 over A280. The A260 is to get the concentration of the DNA based on the fact that all of these nucleotides, because of their aromatic bases, absorb light at about 260, right? It's not the maximum for every base, but it's within range of the maxima for every base. So it's a good approximation of you can actually use this number, this extinction coefficient, and the extinction coefficient of it to actually approximate how much of DNA you have, okay? So you can get the concentration. Okay, so just putting these bases in context in their actual nucleotide and nucleoside forms, this is, again, the nucleotide is when you have the phosphate, right? So again, this is adenine, or this is actually, the nucleotide would be deoxyadenylate. This is deoxyguanylate. This is deoxythymidylate. Okay, I can't say this. Thymidylate. I can say it, but it's like Peter Piper picked a pepper. See, this is the problem. Thymidylate. And this is deoxycytidylate. Wow, that is hard. Which is perhaps why I tend to call it deoxycytidine, but it's actually the cytidylate, because it's actually, in this case, this is the phosphorylate, right? The part in orange is deoxycytidine, or deoxythymidine, deoxyguanosine, deoxyadenosine. These are the nucleosides. Now, we don't say all of these words every single time. Instead, what we say is that this is A, this is G, this is T, and this is C. Right? That is the more common way for us to refer to the bases when we're talking about sequence. So in RNA, it's the same exact thing. The only difference is that these are now the hydroxyl group is there at the 2' position, and this is uridine or uridylate. Okay? So same exact thing, but this is now uracil and not thymidine. So it doesn't have that CH3 group here. Okay. So, these differences in RNA and DNA, what do they matter, right? Like we have these, I showed you that those structures are really different, but like what are the differences in RNA and DNA? Like what are they doing? What do we care about them? So, there are two main differences in RNA and DNA. One is that hydroxyl group, that pesky hydroxyl group. But the second is the uracil, and we're going to talk about what both of those do in sort of a chemical and biological setting, okay? So, the 2' hydroxyl group, what do you think is the importance of this? And you don't have to answer this on your sheet. Just what do you think is the importance of this?
It makes the ribose or RNA more unstable.
Yeah, it makes it more prone to hydrolysis. So it turns out that the phosphodiester linkage in DNA is incredibly stable. They have found DNA that is thousands of years old, right, and still intact. Okay, they think they can get at least bits of DNA from dinosaurs. I mean, that's pretty good, right? From amber, that's actually really crazy. Okay, they're thinking about trying to clone mammoths, and that's because the DNA is that stable. right? What does that mean? It means that you stick it in water for very long periods of time and it's just fine, okay? RNA, on the other hand, is not at all stable. In fact, if you ever work with RNA, you're usually hyper careful to try to keep it from being degraded. Part of that has to do with the fact that there are enzymes known as RNases that are on our skin that actually can degrade them, but part of that is also that RNA is inherently less stable than DNA. And this hydroxyl group is actually why. So the reason is because if you look at base-catalyzed hydrolysis of the phosphate ester in ribose, right, in RNA, you have this ability for this 2' hydroxyl group to perform an attack directly on the phosphate, okay? And so it can go through this cyclic monophosphate derivative, which can then have other things, but what ends up happening is you end up cleaving your RNA. Okay, so this hydroxyl group, because of this base-catalyzed hydrolysis, because it can actually assist, it basically pays part of that entropic cost already, right? Because it's right there. So the probability of getting the right attack angle is much, much higher. Okay, which makes this much, much, much more likely to happen, which means the rate of it is higher. This is not a thermodynamic problem. This is a rate problem, right? This is a probability issue. DNA without this has a lower probability of hydrolysis, right? Thermodynamically, there's not that much difference between them for cleavage, right? But RNA, because this hydroxyl group is right there, once it's depregnated, it can just come in, right? The probability of that attack is now much higher. So you get cleavage, okay? So the hydrolysis of this is much, much faster. This is a rate issue. A second consequence of this 2' hydroxyl group is actually a structural consequence. And so most books don't talk about this, but I think it's worth thinking about, okay? And it's a pretty simple structural consequence. So if you look at this is a 5-membered ring, right? It's kind of like that pentane that we could do the envelope on. If you look at the envelope structure, okay, in the B form of DNA, which we're going to talk about, so this is the double helix DNA. The classic Watson-Crick DNA is the B form. In that form of DNA, it has a conformation that's called C2 endo, which means that the envelope flap is up, and this carbon is up on the same side as the C5 prime. Okay, that's what this conformation is. This is the required conformation for the B form of DNA. Okay, why does that matter? Because if you look at this, so this is our, so let me just get myself oriented here. So this is our 5' phosphate, right? Here is our ring. Here's our here's our oxygen on our 5 position, right? Here's our ring. This is 4. That's C3. This is C2. Okay. If we look at where that hydroxyl group would be, it would be right here. This is the C2 endo form, C2' endo form, right? There's only 1.9 angstroms between this hydrogen here, which is that right there, and this phosphate group here. It can accommodate a hydrogen, which is what you have in deoxyribonucleic acids. Could it accommodate an oxygen and an OH? No, it's way too big. You have steric clash that prohibits this particular conformation from being achieved by RNA. Because this conformation is required for the B form of DNA, which is the double helix that we'll talk about, right? You can't get that exact conformation for RNA the way that you can for DNA. Okay, so there's a structural consequence to having this be a hydroxyl group instead of being a hydrogen that has to do with steric hindrance and steric clash. Only so certain conformations are not accessed. Okay. The second difference is that we have uracil in RNA and thymine in DNA. Why? So to think about this question, we need to think about cytosine. And you're like, why are we thinking about cytosine if we're talking about uracil and thymine, right? Okay, so this is what cytosine looks like. This is what uracil looks like. Can you think of any way by which cytosine could go to uracil? Nothing super obvious, right? But what if we made the imine form of cytosine and then hydrolyzed it, right? What would that product end up looking like? kind of like that, right? So if you hydrolyze cytosine, you can actually get uracil. So if you think about the mechanism, if you made the imine form and then attacked with a hydroxyl group, and then eventually you would get to this, right? This would be the more stable form of that amine. So this happens all the time. It's common. It happens at a basal level all the time in your DNA, right? But it's not a problem. Why? Because uracil is not what's used by the genetic code. So you have checks and balances in your system that can recognize that it's uracil and not thymine, right? Because it doesn't have that methyl group. And it can then remove it. It edits it out. Okay? And because this can go to this, but it can't go to that. Right? Methyl cytosine, which actually can happen, so there are cytosine methylation enzymes in the body. Methyl cytosine can occur. It's not as common as cytosine, right? It's a post-transcriptional modification. This can occur, and in the case of methyl cytosine, you do get thymine if you do that hydrolysis. And this is much harder to recognize, and as a result, this is the most common mutation, okay? Because this does not have as much editing mechanisms. So this is a more common mutation. But this is a lot less common in your DNA than cytosine, right? This is just not present as much. It's another modification on top of the bases, okay? So because of this, so it's an advantage to have uracil in RNA and thymine in DNA because it provides a handle by which your editing enzymes can actually recognize issues in replicating genetic code, okay? So which would you choose to create a genetic code? And why? What do you think? Which would you choose? How many people would choose DNA? How many people would choose RNA? Now somebody give me an explanation for why. One reason. Any reason.
It's more stable.
More stable, right? Because if you want a genetic code, if you want to replicate it through generations, right? So that for thousands of years you have the same genes, right? How can you do that if your RNA is going to hydrolyze? And then the other issue is that it's less prone to mutation just due to hydrolysis of the cytosine, right? So you want something that doesn't have too high of a mutational rate because some amount of mutation in evolution is good, but a high amount of mutation is often bad, right? Because then you mutate things that, say, really matter, like the ability for your cells to use oxygen, or the ability of your cells to make ATP, or the ability of your cells to do a million other really, really critical functions, right? So you have to have a moderate level of mutation. Okay, so I'm going to stop here, but next time we're going to talk about the nomenclature for DNA and RNA, and we'll start to talk about Watson Cricket and all of that. But I do want to point out that there was homework, and it's mostly just reading. Okay, I did not assign any